The next speaker is Dr. Lingjun Li. Lingjun is a Vilas Distinguished Achievement Professor and the Charles Melbourne Johnson Distinguished Chair Professor of Pharmaceutical Sciences and Chemistry at the University of Wisconsin-Madison. Today, she will present on high-resolution FTMS-enabled mass defect-based chemical tags for multiplex quantitative omics.
Thank you, Andrew. Can you hear me okay?
Yes, you're perfect.
Okay. Great, thank you. Thank you, CIL, for the great opportunity to present to you. Also want to congratulate CIL for 40 years of successful business and accomplishment. So today I want to talk about some of our effort on developing chemical tags for multi-omics or quantitative omics. So I'd like to begin by showing this actually probably a very familiar workflow for mass spec-based bottom-up proteomics. So depending on you're working with cell culture or piece of tissue, get the proteins out, and then for digestion and fractionation, with just kind of a nanoflow LC-MS routinely, we can get thousands of protein identification from a single experiment. However, to really understand the biology, oftentimes we need to get more quantitative information. So within mass spec-based quantification strategies, there are two big categories, either absolute quantification that rely on building calibration curve, or relative quantification where you want to compare multiple conditions. And this can come with label-free or chemical derivatization approach. Since this is an isotope day, I thought it's actually pretty appropriate to focus on discussing stable isotope labeling approaches. And so within this, again, two big categories, the mass difference labeling, oftentimes they rely on either incorporating light or heavy isotope encoded for metabolism, metabolic labeling, or chemical labeling. So you have a MS1 based quantification, look at area under curve. And alternatively, people increasingly turn to isobaric labeling. So this could rely on a set of chemical tags that give rise to the same mass. And then in the MS1 mode, they show as a single peak, so there's no spectral complexity involved. However, the MS2 allows the production of reporter ion for you to get relative quantification for multiple samples, and MS for identification. So there are two actually very successful commercial products, such as TMT and iTRAQ. So our lab has been actually spending quite a bit of effort to develop a cost-effective alternative focused on dimethylated leucine or dimethylated amino acids, which would be the focus of today's talk. So there are actually a number of actually key advantages to increase multiplexing. For example, this would help us to reduce the amount of any one sample for your analysis. And this would also translate oftentimes to reduce sample preparation or analysis time. And this, if you're using facility instrument, the saving of instrument time is actually a great bonus. And also increasingly, we're asked to look at a number, actually a large number of biological conditions or replicates in a single experiment. And finally, this would also help to increase the LC-MS data overlap between conditions. So our first generation of dimethylated leucine tag shown here. So here is a amine reactive group, a triazine ester, and the carbonyl group, balance group, and then the dimethylated leucine. So it's very similar to either iTRAQ or TMT based design. And here, by incorporating different stable isotopes, we can create here a plex of 4-plex Dilutex. And showing here in the, for example, we can differentially label 4 sets of samples, combine them in MS1, so there is no spectral complexity involved, and then MS2 allows us to break these two bonds to generate reporter ion ranging from 115 to 118, is 1 Dalton apart from each other, so we can quantify their relative abundance from 4 samples, and then the backbone cleavage for identification. So in order to further improve our throughput, we turn to this phenomenon called isotopic mass defect, which is described by this Einstein energy equation. As we know that the light and heavy isotopes from the same elements that bind to the neutron energy difference that could lead to a small, tiny mass difference. For example, if we swap deuterium with carbon 13, this would give rise to 2.9 millidalton mass difference. Likewise, if you substitute C13 with M15, there is a 6.3 millidalton difference. So based on this idea, Dustin Frost in our lab has designed these 12-plex DiLeu isobaric tag reagents. So based on the isotopic mass defect, but also the access to the high resolution Orbitrap type of instrument. For example, if you look at the 115 A and B, if we substitute the M15 with this carbon 13, and this would have a 6 millidalton mass difference. So based on this, we can actually construct this second generation of Dilutex. We're still using the same chemical structures. It's a very compact structure, only require 1 or 2 step synthesis, and there's no need for any custom isotopic reagents. So we can keep the cost down and also produce a large quantity. And showing on the right is the isotopic configuration. As you can see, we can use O18 or carbon 13 as the balancer to generate this 12-plex Dilutex. So you may be wondering what kind of resolution that is needed to resolve these 12-plex channels. So we have actually systematically looked at a number of these different mass spectral resolving power from 15K to 240K. As you can see very clearly from at 30,000, we can actually baseline resolve these 12 reporter ion channels, so that basically it can be achieved on most modern mass spectrometers, Orbitrap for sure, but even some of the high-end Q-top systems. So just give you an idea of using this 12-plex Dilutex, we look at a tryptic digest from E samples. As you can see in the modest resolution, this would show these 4 peaks, but then when you zoom in with resolving power 60,000, we can clearly resolve these 12 channels for quantification, and the backbone cleavage gives pretty confident identification for tryptic peptide. We then evaluate the large scale for combining 1-to-1 ratio and also 16-to-8 to 4-to-1 ratio with over thousands of these proteins. They all show pretty good quantification performance. So then we ask ourselves the question, can we go even further? So we note that if we can shrink the mass difference between the adjacent quantitative channel, for example, to 3 millidalton, we can actually design just 21-plex Dilutex. So showing here again, just remind you, this is the same compact chemical structure. And still, we're very actually happy that we don't need to rely on any custom design isotopic reagents, so just use the commercially available amino acid isotopes readily available from Kindred Isotope or Sigma Aldrich. As you can see on the right-hand side, this is the isotope configurations. And if you look at a little closer, we have added additional 9 quantitative channels. So what we notice is that here, instead of doing the pair dimethylation, here we actually just need to use the monomethylation strategy to introduce odd number of deuteriums, either 1 deuterium atom or 3 deuterium atoms. At 115 and 116, it requires an O18, an extra step of O18 exchange. So based on this, we have designed an monomethylation synthetic strategy where we actually partially protect the primary amine using this chlorobenzyl formate, and then so generate this secondary amine to react with the heavy isotope aldomethane. And then this would incorporate, for example, in this case, a single deuterium atom, and then simultaneously deprotect and react with formaldehyde doing the regular methylation, adding the second methyl group. And then in this case, we can use O18 exchange to add this balancer. So here, as you can see, with either 3 or 4 steps, we can actually synthesize these 21-plex Dilutex. So then the question is, again, how much resolution that is needed to resolve them. So if we look at the adjacent quantitative channel for the 115 A, B, and C, and the smallest mass difference is the 2.92 millidalton between the 115 A and B, and this can be actually baseline resolved with 60,000 resolving power. And showing on the bottom is the 21 quantitative channels can be readily resolved with 60,000. So for example, for 118 reporter ions, now we have 7 variants and 6 variants for 117 and 5 of these for the 116. So that helps us to actually boost up the quantification throughput quite a lot. So then just to show one representative tandem mass spectra to show the quality of the data, and this is taking K56 to triplet digest. As you can see, again, in the low mass region at 60,000 resolving power, we can actually quantify these 21 channels. So here we combine these in 21 ways in 1-to-1 ratio. And here again, the B and Y fragment ions allows us to confidently identify this particular triplet peptide with X-core value of 4.7. So collectively, we want to combine and look at, evaluate the proteomic performance of this 21-plex Dilutex to see, for example, at the peptide identification level. So with the blue bars, these the DiLeu label versus the unlabeled. As you can see, we have actually pretty good X-core value, and that shows the high confidence, and also the peptide sequence primarily ranging from 6 to 10 amino acid residues, triplet peptides. And this also induced a little bit enhancement for the charge state, and the quantification rate at the protein level achieved at 99%, and peptide spectrum match at 95%. We've also evaluated its quantification ratio at 1-to-1 and 15-to-1, and this different combination ratio all show pretty decent, acceptable quantification performance. And this last slide panel shows the average reporter ion signal to noise ratio. So next, I want to tell you 2 application examples. The first example I want to tell you about is to use the Dilutex to combine proteome and metabolome analysis on the same analytical platform. So here we're looking at pancreatic cancer cells. We can extract proteins and metabolize separately. And here introducing the Dilutex, this would actually help to increase the hydrophobicity for some of these metabolites. Obviously, we're targeting the amine-containing metabolites. So as we know that typically metabolome analysis is done on the regular flow LC MS. In this case, we want to use the same analytical platform, which is the nanoflow LC system. So here shows the mass defect-based Dilutex. As you can see, the light label, here we have the N15, carbon 13, and O18, and the heavy label with 4 deuterium atoms. So the 2 channels is differing by 20.5 millidalton. So this small mass difference can be resolved with high resolution Orbitrap type of instrument at MS1 level. So this would just based on the area under curve. And so you may again wonder what kind of resolution that is required. So for MS1 based centric quantification at the protein level, we actually observe at 240,000 resolving power is needed. And for metabolites, 120,000 resolving power is required. So another actually benefit is that using the Dilutex, we note there is actually a substantial boost up of the sensitivity. As you can see in the blue trace, these are labeled cellular metabolites compared to the green. We actually get a pretty substantial increase in the signal intensity. And also this helps to increase the retention and also separation. So the bottom is the non-derivatized isoleucine and leucines that are partially resolved after dilute labeling. You can see they actually have very well separated and also increase the retention time. So then we want to evaluate the MS1 based and MS2 based quantification. In this case, the box plot and also the linear dynamic range shows pretty comparable performance for both of these, with MS1 shows slightly better quantification accuracy. And then we want to look at both the protein and the metabolite, their quantification accuracy and dynamic range. Again, we actually observe pretty comparable for both approaches. So our goal is to combine these 2, for example, using functional enrichment analysis to look at both the cellular components for the proteome and metabolite, as well as the biological processes. So here we can actually combine the two and identify 7 of these pathways that overlap between the proteome and metabolome analysis. So this gives rise to, for example, some of the very well-known metabolism pathways such as glutathione or arginine proline metabolism pathway. So to kind of summarize this part, I hope I've shown you that we can use the mass defect Dilutex to perform MS1-based quantification in parallel proteome and metabolome on the same nanoflow LC-MS platform with improved quantification accuracy and detection sensitivity. So the second and last application I want to tell you about is the use of Dilutex to look at biomarker discovery in Alzheimer's disease. So AD is perhaps one of the most best known neurological disorders and is ranked number 6 leading cause in the US and also worldwide. Currently, more than 5 million American people live in this condition and there's no cure. So clearly, if we can develop early diagnostic assay, this will help tremendously. So this work was done in collaboration with Professor Cindy Carson and Ozima Nkonko at Wisconsin ADRC. As we know that Alzheimer's disease is a progressive neurodegeneration, can be separated and divided according to different stages, preclinical, mild cognitive impairment, and dementia based on biochemical, anatomical, and clinical functions. So these different stages actually can span multiple decades. So if we can develop early biomarkers that will help tremendously in terms of diagnostic and early intervention. So using actually non-targeted proteomics approach, we have looked at large number of proteins from cerebrospinal fluids and showing here are some of the candidate biomarker that we like to further validate. So showing here are 3 of these examples, apolipoprotein E that is known to modulate amyloid beta aggregation and clearance, and also transthyretin that's known to bind to beta amyloid protein to prevent it from forming plaques, and VEGF that is known to promote survival of neuron. So one of the golden standard to validate some of these candidate biomarkers is obviously targeted proteomics using either SRM or PRM type of experiment. However, there's still some limitations related to this type of experiments in that there's low specificity in complex samples, but more importantly, the limited analytical sample throughput, and also less accurate estimate when the amount of target peptides stand for wide dynamic range, which is common the case for biofluids. So to address some of these challenges, again, we want to turn to stable isotope labels. As I already introduced about the isobaric labeling, I want to briefly introduce the mass difference labeling for still based on the same chemical structure of our DiLeu. Instead, here we are actually introducing different number of deuterium atoms to create a 5-plex isotopic DiLeu or iDiLeu that is differing from each other by 3 Dalton in the adjacent channel. So this would allow us to actually use, for example, 4 of these isotopic DiLeu channel to label the known concentration of target peptides and then perform the absolute concentration. So when we perform and when we look at these 2 sets of tags, we know that the 12-plex Dilutex actually has a 145 Dalton mass addition, which is very similar to the D3 channel of the iDiLeu. So we wonder whether we could actually substitute the D3 channel with the 12-plex DiLeu label, for example, CSF samples, to increase the quantification throughput by 12-fold. So based on this idea, Xiaofang Zhong has designed this hot max strategy where we turned hybrid offset triggered multiplex absolute quantification. So the idea is that we can use the 4 iDiLeu channels to label our target peptides with serial dilution and then spike into our 12-plex DiLeu labeled CSF samples. Because the iDiLeu and DiLeu share the same chemical structure, so they will co-elute in the LC-MS profile. So this would allow us to perform this internal standard calibration curve absolute quantification. Furthermore, this D0 iDiLeu labeled standard can actually act as a real-time trigger. We can perform MSMS, and when it's actually matched the inclusion product mass list, it will trigger this 4 Dalton mass offset, trigger the 12-plex labeled target peptides to perform this MS2-based relative quantification. And to further improve the quantification accuracy, we've also introduced this synchronous precursor selection MS3 approach to further reduce the quantification error. So then we want to evaluate the feasibility of this HotMac method. So here we have a iDiLeu D0 labeled TTR tryptic peptide to spike in the 12-plex DiLeu labeled CSF samples. And here, 2 of these cluster peaks shows, again, 4 Dalton mass shift. So we can use the D0 labeled peptide to trigger this real sample, 12-plex DiLeu sample. As you can see, they show identical mass. And this would allow us to actually quantify all 12 channels and look at the quantification accuracy with 1:1 and 1:2 to 5:10 ratio. And also we want to compare our HotMac method with the more established SPS-MS3 and PRM-MS2 approach. As you can see from this radar plot, the HotMac approach actually shows the smallest relative error for compared to these 2 methods at all these 3 different combination ratios. And showing at the bottom is the linear dynamic range and also the absolute quantification. So finally, we just want to use this to look at the real cerebral spinal fluid samples collected from 22 patients. So 11 preclinical AD and then 11 healthy control. As you can see, these 3 targeted proteins that show pretty good linear dynamic range. And the bottom shows that the ApoE, for example, in this case, shows downregulation with statistical significance. So the final couple minutes, I want to briefly
John, you're down to the wire.
Yeah, I'm wrapping up. And
Okay.
the glycosylation in Alzheimer's disease, in particular, we want to use our Dilutex to look at glycoproteome. So here, the protein glycosylation has 3 different levels, changes in either protein abundance or the glycosite occupancy or these glycoform change at site-specific or microheterogeneity fashion. So in collaboration with Wisconsin ADRC, we have developed this integrated analytical workflow where we can look at the glycoproteome from cerebral spinal fluid, but I want to just focus on this Dilutex, the use of this Dilutex to quantify these glycoproteome from 48 patient samples divided in 3 groups, the control, MCI, and AD, and we were able to quantify over 1,500 N-glycopeptides amount to 107 N-glycoproteins. So this volcano plot shows that actually the dysregulated N-glycopeptides, for example, 69 of these showed changes in the MCI and 132 dysregulated at AD condition. With gene ontology analysis, we actually reviewed the top ranked processes, include platelet degranulation and cell adhesion and others. So the final piece is to look at these N-glycopeptides. We picked 19 of these that show consistent changing trend in MCI and AD. So you may be wondering whether these quantifications happening at the protein level or at the glycoside. So here we took one of these apolipoprotein D that actually does not show significant changes at the protein level, but at these 2 N-glycoside 65 and 98 shows actually pretty systematic changes here. And there are 8 of these N-glycopeptides show upregulation, and then the 98 position, which occupies at the coil junction of these, showing downregulation. And we have looked at the microheterogeneity of these different glycoprotein. And given the interest of time, I just want to pick out one of these. Clustering is the 3rd most risk gene in AD, and there are 4 different glycosides. Some of these have very modest microheterogeneity. However, at 374, we observe 56 different glycoforms. So we can actually also use the N-Dilutex tag to look at these AD stage specific N-glycopeptides. For example, we can look at specifically at MCI, some of these peptides show upregulation, and at the AD condition, most of these actually show downregulation. So to kind of wrap up, I hope I've shown you that we have developed a suite of these dimethylated amino acid tags that can take out different protein quantification depending on the purpose, either for absolute quantification or relative quantification, depending on the number of samples and that you are interested in looking at, and also looking at MS2 or MS1 based quantification. I also want to thank the P41 Center under the leadership of Josh Kuhn to support some of this work that I talked about today. And also want to thank people in my lab that showed their pictures, and also the collaboration and funding, and all of you for your kind attention. I'd be happy to answer any questions if there are any for time. Thank you.
Thank you, Lingjun, for an excellent talk. The multiplexing quantitative potential of these isobaric tags is truly impressive. Unfortunately, we need to move on due to time constraints, but thanks again.
Thank you.
I'd like to